Today, we will be going over the cell signaling pathway of acetylcholine. Acetylcholine is a hydrophilic signaling molecule. It uses membrane integral proteins called ligand-gated ion channels to communicate. Ligand-gated channels are mostly responsible for synaptic transmissions in the nervous system. In the case of acetylcholine, they are responsible for activating a pathway that leads to muscle contraction. Ligand-gated channels work very rapidly, but the signals are short-lived and do not last long. Acetylcholine is a neurotransmitter that is responsible for many functions in the body, such as muscle contractions and memory. There are two types of receptors for acetylcholine, muscarinic and ionotropic. Muscarinic is responsible for the contraction of involuntary muscles and operates with the G protein coupled receptors. Ionotropic receptors are responsible for the contractions of voluntary muscles and operates with the ligand-gated channels. Today, the focus will be the ionotropic receptors only. The signaling pathway starts when acetylcholine is released from the presynaptic neurons in the synaptic cleft. It then binds to the ionotropic receptors on the surface of muscle cells. This alters the shape of the ionotropic receptors and produces a small passageway through the protein for the sodium ions to go through. As sodium ions flow into the cell, the concentration of ions inside the cell change rapidly, causing depolarization inside the cell membrane of the postsynaptic cell, meaning that the inside of the cell becomes less negative than the resting cell membrane potential. Once depolarization reaches a certain point, it triggers an electrical impulse in the postsynaptic cell. This action potential can propagate or travel along the muscle cell, leading to the transmission of this signal, which leads to muscle contraction. The signal is terminated when acetylcholinesterase, an enzyme in the synaptic cleft, breaks down acetylcholine back into acetate and choline, which are both recycled.